talk. Up next is my friend Mahmoud Ghannoum, who's going to give us an update on the fungal mycobiome. This is a topic he talked about it in the last biennial meeting 4 years ago, and I'm very curious to see where we are now.
Well, first of all, thank you very much and for inviting me to give this talk. And I tell you, it's a great pleasure to see all the good friends in these meetings. We miss it so much with COVID, so it's a great occasion. So I wanted to look at the fungal microbiome, especially when it is under the consideration for future trials. And this is my conflict of interest. First of all, I'm a co-founder of Biome Health. It's a company that develop probiotic and do gut testing. Also, I receive funding from most of the companies that do antifungals or developing antifungals. So you can see all those. What I would like to do in this talk is basically I want to show that the microbiome gaining traction. And the microbiome, it means the fungal community, okay, just for those who didn't figure it out so far. Next, I want to show that the microbiome also, how it affects disease, and take an example in Crohn's disease. And thirdly, I would like to talk about using the microbiome data. You listened about big data, so I'm going to tell you how we use this microbiome data to develop products and so that we can rebalance and maintain the microbiome harmony. And finally, how do I think we should use the microbiome in future clinical trials? To start with, whenever I used to go to a meeting about the microbiome, everybody talking about bacteria. That really irked me a lot, especially since I did my PhD where effect of steroids and antibiotics on Candida. And I tell you, at my age, it was over 46 years ago. And I knew that it is not only bacteria, because once you kill the bacteria, by the way, using antibiotics, I'm going to show something, guess what? Candida start to grow. And that's why I think we need to focus on the microbiome as well. And of course, I didn't stay still. I wrote 2 articles, one in the microbiome in 2010, telling them about the microbiome as a really ignored kingdom. Nobody heard. I went again 2016 in The Scientist and wrote an article, really called it the microbiome, the ignored kingdom, and told them this story. And you can see in this, my left hand at least, this is the number of publications in 2015. Only a handful of them was on the microbiome. Okay, the vast majority, over 700 articles were published on the bacteria. So then, to my delight, last year, people start to figure it out, including the NIH, where they wrote that an article, an IDCR, saying that the microbiome beyond bacteria. And I was invited to give a talk in the Forsyth Institute, and this is the title of the meeting, Oral Microbiome Beyond Bacteria. So it became clear that it is not enough just to look at the bacteria. You need to look at the fungi, viruses, and this sort of thing, because this is very, very important. So it only took them about a decade to figure that out. Now, the other important point which I would like you to know is that both bacteria and fungi, they are living in a sandbox. They interact together. Like when babies or children play in a sandbox, if they are happy, the parents are happy, and if they are not, then guess what? We all suffer from that. And that's why I wrote an opinion piece in MBio talking about really the cooperation between fungi and bacteria, and this was from a study showing that wound where both bacteria and fungi form biofilm, and in order, as you know, to really break through, you need to dissolve this matrix, and then you will be able to do something about it. Okay? Now, I would say to you that the microbiome has gained much attention, and these are just some papers which we recently published showing you that the microbiome can impact our health, especially the gastrointestinal tract as well as cancer. And so this is, I like you to start looking at these articles because it's a lot of studies. And the good news also, the last 2 here, articles published in 2022, just looking at what is the change in the microbiome in cancer patients. And we were able to show that really fungi is involved intimately in this process. So please, you need to look at both the babies, okay, and the mother. And everybody tells us, you know, bacteria are so many trillions. I agree, but look how big. I should go back. How big is the fungus? Here is Candida tropicalis. 12 different bacteria had to attach to it. So size matters, my friend. Now, I want to go into the using the microbiome data to design new products, and I want to take this as an example here from the stool up to a product, okay? And taking Crohn's disease as an example. Here we looked at a family with really patient with CD and their sibling or people in the same family with no Crohn's disease, and also we looked at control. Anyway, we then analyzed their microbiome, both bacteria and fungi, and what we found that in Crohn's disease patients, you can see here CD versus non-CD, we started to see really a decrease in Saccharomyces cerevisiae, which is a good body. It's a friendly yeast, as you know. In healthy people, it really high compared to the other one. But also, we found statistically significant increase in Candida tropicalis. In addition to the Candida, we saw also a decrease in Faecalibacterium, which I don't know how to say the second name. It's an anti-inflammatory bacteria. It goes really down, while we see an increase in E. coli and Serratia marcescens. And when we did correlation analysis, we found that there is very good correlation between the Candida tropicalis and these E. coli and Serratia. And to our mind, as a person who worked in biofilms in the gut, these organisms, they form biofilm. So we said, let's look at that. And here is the Candida tropicalis alone. You can see yeast. This is Candida and E. coli. It really fused to the yeast. And then when you put the three together, look what happened. It changed the form, okay? So that's in vitro. We also looked in vivo, and this is the quantitation of this observation. But anyway, this is in DSS, dextrose sodium sulfate model, which is a Crohn's disease model. You see, this is DSS only. This is with the three organisms, and you can see a huge biofilm forming there. And of course, as you can imagine, this start to cause invasion of the gut lining and cause, guess what? Leaky gut, most probably. So we published this paper, and I think it really was very well received, okay? Now, I want to tell you, look, when you have Candida tropicalis, you have issues in the gut in this Crohn's disease model, okay? It's a colitis model. So the first thing you can observe is that if you have a oral challenge of the mice, you will have more death in those infected with Candida tropicalis. Also, you have changes in the structure of the villi, where you see more inflammation and thickening of all the villi. And really, if you look here, this is not infected versus infected. This endoscopy, clearly the tropicalis cause issues there. We also looked at their microbiome, and there are changes as well there. This is at baseline, and then after you challenge, you see there is the profile start to change of the microbiome, particularly with respect to Akkermansia, okay? Now, we also looked at some immune factors, and you can see some of these are statistically significantly increased. For example, here, interferon gamma, as well as you will see some of them decrease, such as IL-4, IL-13 as well. So there is effect in the immune system as well. So in summary, having Candida tropicalis is an issue in these Crohn's disease patients. And that's why we really need to find ways to try to address this, okay? The first, what I want to talk to you about is really talking how we can find a probiotic that can rebalance the microbiome, and then later on how we can use an antifungal to really help us in making these patients better. So for us, with the data which I showed you in the Crohn's disease families, we wanted to identify strains that are really antagonize E. coli, Serratia, as well as Candida. So that's the first step. But the other thing, because we have biofilms, we added an enzyme amylase, which have been shown to have anti-biofilm activity. So we wanted to break the biofilm, also antagonize these pathogens. This is how we did the correlation analysis. I'm not going to go into details, but you can see here in red, Saccharomyces and Candida antagonize each other. So having Saccharomyces boulardii, for example, is a good way to include in whatever probiotic you want to do. And this is some of the studies. We formed a probiotic which has 4 different strains. This is untreated biofilm in vitro. This is treated, and you can see really eliminated the biofilm, and the same thing we could see in the in vivo. Then we did a clinical trial to evaluate whether this probiotic which we designed, can it have effect on the microbiome? And to cut long story short, these are the 49 subjects were analyzed. We took at baseline and after treatment, and we looked at their microbiome. And what we found is the following. Number one, we started to decrease zygomycota was high in those Crohn's disease patients, and in the patient where it went down. And also we had a significant decrease in the abundance of Candida. So this probiotic is doing something. Then I'm not going to go over detail with this. Basically, it was positively impacting the microbiome where we decreased, particularly I'm focusing here, the Candida. We also just finished a clinical trial, which is a randomized placebo-controlled double-blind trial where we compared people on this probiotic for 6 weeks versus the placebo. And you can see we really met the primary criteria. It was submitted to Frontiers. It's under revision now, so hopefully it will come out soon. But the primary criteria was abdominal pain, flatulence, and bloating really were significantly reduced with the people on this antibiotics. We also looked at the microbiome. Also, it adjusted it in such a way that we have basically increase in the abundance of Saccharomyces, more than 200-fold increase, which, as I mentioned, this is a really beneficial organism. So this is the conclusion of basically the clinical trial. We met the primary criteria. There was other secondary criteria, such as constipation, also was successful, and we used GSRS scoring, which really takes all 15 different GI symptoms, and that was also reduced in these patients. So now you can see, I took you from the beginning, the sequencing, we all the way in vitro, the correlation analysis, the biofilm, we published that, and then we did these 2 clinical trials. So this is big data, my friend, going into to help you in that. So now the last part, which is really relevant to this meeting, is the microbiome and future clinical trials. So I look at the situation. I thought we really need 2 ways to try to improve the Crohn's disease issues. Number one is, I think by the end of this, I will know how to use this, but it's difficult. Number one is the antifungal. I think it will be important to use where it will eliminate Candida. Also, we know some of the antifungals, they have very good anti-biofilm activity. Whereas in the probiotic, why to have it? Because number one, we showed that it affects the biofilm. It also produced short-chain fatty acids, which I did not show you data, but we have to show data to show that. Also, it increased the level of beneficial organisms, and finally, leads to significant reduction in GI symptoms. So this is how I look at what we really should do. We need to have potential synergy between antifungals and probiotic, okay? Number one, in the antifungal, you eliminate the bad guys, then you stabilize it with the probiotic, and then you need to maintain it, okay? Now, one last point which I want to bring into your attention before I finish is we would like to have clinical trials to assess whether an antifungal affect or is affected by the microbiome. Now, if you take into consideration the fact that drugs, a good example, what my friend talked to you, Contayanis, about, guess what? The checkpoint inhibitors. 30% of people on checkpoint inhibitors fail therapy. You know why? Because the microbiome have been shown to break down that drug. So we need to know what is the effect of drugs on the microbiome. And number two, can the microbiome affect the efficacy of the drug? Okay? And recent concerns, now it's becoming, people are starting to ask, both consumers, patients, as well as there are FDA starting to ask, oh, is this drug good for the microbiome? So because of this, I propose to MSG to start thinking about medical mycology committees should include specific aims to try to evaluate, to be evaluating in the clinical trial to answer whether the antifungal under investigation affects or is affected by the microbiome. And I think this is a study we just published in March of this year where we evaluated broad spectrum versus narrow spectrum antibiotics, and guess what? We found differences between them, and we selected representative organisms that are usually present in the gut, and we did MIC for them, and guess what? The narrow spectrum antibiotic, all these arrows showed that it has less activity against this compound against these organisms compared to the broad spectrum activity. So there is already ways to do this both in vitro as well as in animal models. And these are the people who really did the work from our Center for Medical Mycology and Integrated Microbiome, Tom McCormick, and really Helmi, who is the bioinformatician, and Mauricio, who is the sequencing guy. You have people, we worked with the GI Institute at Case, and we have funding from the national, I have R01 to look at the interaction between bacteria and fungi from NIH, as well as Biome Health paid for the studies related to the probiotic. And this is an advertisement. This is a good open access journal. You don't have to pay for publication. It's from Case Western Reserve University. It's really, I recommend it. I'm biased, of course. So send your papers there. Thank you.